All right, we want to deal with a few other topics having to do with embryology. We're kind of finished with our different drawings, showing where the different parts come from. Now, first term we want to talk about here is parturition. That's another word for birth. Just want to bring that up because the next terms have a portion of this word in it. The first one being ovipary, or the oviparous condition. This refers to things that basically they give birth to eggs, the egg layers. So that's how they get their start. The other term related to that viviparity has to do with giving birth to a live organism, so live birth. Give birth to living things that are already in a more advanced state. Live birth versus birth to eggs. Okay, so that's our first comparison. Under this category of viviparity, there are two different conditions. Ovoviviparity, live birth of the young after development of yolked eggs that were retained in the mother. So it was, imagine a chicken that instead of laying eggs and having them develop outside of the body, the mother retains them inside the body, and over time they go through their entire development, and when they're about hatching size, the mother gives birth to an already hatched kind of egg. These don't develop the shell, obviously, that you'd have in an oviparous type of organism, but anyway. So they don't have a placental type development. The nutrient delivery to the developing embryo is by the yolked egg, not by a placenta. Now the contrasted term to ovoviviparity is typically euviviparity, euviviparity. Live birth of the young, which were nourished by placental development. The young were developed in the mother like these, only in this case, instead of having a yolked egg to provide nutrient, they had placental nutrient delivered to them. So nourishment from the mother delivered across some kind of placental boundary to the developing young. Now regarding some examples of these different birth types. First, oviparity, laying eggs. That's not a very uncommon condition. We're all familiar with that arrangement. Most fish, amphibians, reptiles, and all birds lay eggs. They deposit the egg develops outside of the mother's body and hatches later. Among the viviparous types to give live birth, the ovoviviparity example, things that do this include some fish. Some fish give live birth. They retain the young in the body until they develop. Guppies is a common example there of a live birth group in the teleos, but even in the coelacanth, you see this. Some of the sharks retain their young in the body until they're finished developing and give live birth. Many of the sharks do that. So this is a fairly common mode of reproduction as well, giving birth to young that have developed from a yolked egg. There was a nice big yolk there for it to develop from and undergo its developmental process. Not much nutrient delivery from the mother's tissues. So anyway, they develop inside the body, and then when they are fully matured, ready to to be birthed, they are birthed. Euviviparity, of course, live birth, nourished by placental development. This is pretty much restricted to the mammalian types. Both marsupial organisms and placental organisms have some sort of placenta in there. A kind of slight placenta might be expressed in some of these ovoviviparous types, where some nutrient is given by the mother to the developing young in addition to that material in the yolk. So there's a little bit of variation there. The next thing we're going to talk about are placenta types, variations in the types of placentas in different organisms, and it has to do with primarily their blood supply. Okay, next we're going to talk about placenta types. The three kinds of placenta to note. First one is called the yolk sac placenta. Yolk sac placenta. In this case, a small animal, it's a shark. This is the best example of this. Here's a shark developing here, and it has attached to it a yolk sac. So extending from its belly region here is a large yolk right here. So there's yolk, and it has a blood vascular system that supplies blood from the heart around through the dorsal aorta to the body and gives off vitelline arteries that reach out into this yolk space here, actually around the outside of that yolk that's shown there. So you have an investment of blood vessels retrieving nutrients from that yolk to help that shark body grow, that young shark body there, okay? So this is a yolk sac placenta if this is retained in the mother's body. There are some of the chondrichthyan fish that lay their eggs, deposit eggs in little packets that have the yolk and everything, so they develop outside the body. But in many, this is inside of the mother's uterine tube. So here in that case, if it were wrapped up in a uterine tube, let's see, let's put a uterine tube around the outside of this little baby shark. So it's in this tubular passage here. There could be blood vessels from the mother shark here adjacent to this yolk, so you get nutrient delivery across from the mother's uterine lining to the blood vessels of that yolk that's there next to the developing shark. So in that case, you would have a little bit of nutrient delivery in there. That's a yolk sac placenta. The next example choriovitelline placenta, choriovitelline placenta. In this case, we have an embryo. Surrounded by an amniotic cavity in this case, so these are amniotes. It has a foregut and a hindgut with an allantois and a yolk sac out here. And surrounding that, it has that chorionic membrane that's made partly of trophoblast cells, cytotrophoblasts and syncytiotrophoblasts and such. So here, blood vessels move the same pathway around the pharynx and then into the dorsal aorta. We have some blood vessels that go to the yolk sac portion here and out to the chorion. So these are vitelline vessels going to the yolk sac. So this is considered a choriovitelline type of placenta, where we have vessels very similar to these in the shark, where we have that kind of connection But in this case, we have all the chorionic membrane around that, as well as the yolk material. So anyway, we've got vitelline arteries that go out to the chorion, and the mother's tissues would surround that. So this is inside the mother's body, and this would be found in marsupial animals. Marsupials. Now for our third type, let's move the camera a little bit. Chorioallantoic. In the chorioallantoic placenta, again you have your embryo inside of an amniotic cavity. You have your endoderm, which extends from the foregut back to the hindgut, and here we have an allantois that reaches out, and here's our yolk sac. Around the outside we have our layers of trophoblast cells, and in this case, instead of having the blood vessels of the vitelline vessels, go to the yolk sac be the primary vessels to the chorion, it's the allantoic vessels, and that's why we call these the umbilical vessels in true placental mammals. So all of your true placental mammals, your non-marsupials, non-egg-laying mammals, would have that kind of a placenta, chorioallantoic. So it uses its allantoic vessels to come out here and form that placenta. So chorioallantoic placenta here, and we saw choriovitelline placenta in the marsupials, where the vitelline vessels came out to the chorion to be the primary vitelline, the primary nutrient gathering vessels. So a different kind of placenta between the marsupials and the true placentals, the chorioallantoic type placenta. So those are our three placental types: yolk sac placenta, choriovitelline placenta, and chorioallantoic placenta. Remember, your shark is not an amniote. It doesn't have the amniotic membranes, it doesn't have the chorionic membranes, none of that. So it's a different arrangement there with just a yolk sac with vitelline vessels in close contact to the maternal tissues. Okay, so there are some things that are kind of in between, I guess you would say. Most of the reptiles that are amniotes here, they are egg layers, but there are some that retain the eggs inside, like a rattlesnake or many other kinds of snakes, retain the eggs inside the body as they develop, and they could deliver nutrients to the embryo in there, but usually do not. So they are simply ovoviviparous, so they're retained inside, no real nutrient delivery across a membrane, the chorionic membrane in those cases, in the amnion. For our next topic, we're going to be looking at the derivatives of ectoderm, endoderm, and mesoderm. So we'll kind of break it down, and we'll look at what tissues of the body, when you get your final anatomy formed, all the organs in there and everything, what these different tissues contribute to. So let's kind of follow them on out. So the first one here will be ectodermal tissue. You have your general ectoderm that forms on the outside of the body, and we get from that both intraembryonic and extraembryonic. Let's look at the extraembryonic first. Extraembryonic ectoderm can become either a part of the amniotic membrane or in some cases the chorionic membrane. Mainly we're looking at the amniotic membrane and the outside of the umbilical cord in some cases. So we have either amniotic ectoderm, or in some cases like the chicken embryo, we refer to the chorion as an ectoderm. Some might consider trophoblast ectoderm, but not generally. So that's part of those extra embryonic membranes. Remember, we had two membranes that were considered somatopleural membranes, so they include somatic mesoderm and ectoderm. This is the ectodermal portion from those. The rest is embryonic. Ectoderm, and we can break that down into different sections as well. This, first of all, there's surface ectoderm. And that becomes primarily the epidermis of the skin and all things formed from the epidermis of the skin. Hair and nails, for example, your sweat glands, those sorts of things form in the epidermis or from the epidermis. So we have the epidermal component. Next, we have what we call the neural crest component. Neural crest ectoderm. Remember, neural crest wasn't even present in like a amphioxus kind of structure. It's a later developing thing. You only see that in the craniate groups. So anyway, neural crest ectoderm, but it becomes a lot of different things as the animal develops. One of the primary things it becomes is part of your peripheral nervous system as ganglion cells, around those ganglion cells there are what we call satellite cells, the Schwann cells that wrap around your peripheral nerve fibers, so those are all peripheral nervous system components derived from this. Also, cells in your adrenal medulla, that produce epinephrine and norepinephrine as part of your fight or flight response in your adrenal medulla. In your carotid arteries heading to your head, there are some cells that are responsive to both pressure and levels of blood gases. So carotid body cells, we'll call those. Another example of neural crest tissues. In the head region of the developing embryo, usually up in front of the somites, we're talking here, remember somites are mesodermal, so in front of the somites where you don't have any somite mesoderm forming, a lot of your muscle and bone and connective tissue of the head, muscle, bone, and connective tissue of the head is derived from neural crest ectoderm. And we generally just refer to that as head mesenchyme. Mesenchyme is a precursor kind of tissue. It's not fully differentiated into muscle, bone, or connective tissue at that point, so we call it mesenchyme. So those are neural crest derivatives. So we have surface ectoderm, we have neural crest ectoderm, and then we have neural ectoderm. Simply forms your central nervous system. Your brain and spinal cord are derived from neural ectoderm. So these are all your derivatives of the ectodermal layer. Extra embryonic tissues, and then from the embryo, surface ectoderm, neural crest ectoderm, and I should say neural tube, probably there, neural tube ectoderm. Neural tube ectoderm becomes the brain and spinal cord components. All right, next we're going to talk about the endodermal derivatives. Remember that it forms often as either part of a hypoblast layer or cells that move off of a primitive streak and into that hypoblast layer. One of the first things that you often see develop from the endoderm in this fashion is a yolk sac. Part of the yolk sac wall. Remember, it's part mesoderm and part endoderm. So that inner lining of the yolk sac is from endoderm. The remainder that we're concerned with here is primarily from the gut, formation of the gut. And remember that the gut has a foregut, a midgut, and a hindgut. So we'll first look at foregut derivatives, things derived from the foregut region of the animal. The first one to mention here is the pharynx. The pharynx is the most anterior part, not your oral cavity. It comes really from the ectoderm around the face development. So the pharynx. Portions of your respiratory system in the fish, the swim bladder would be that structure, but we'll just identify respiratory passages. I'll just put respiratory, so you know that the tubes and the glands that are along those tubes are derived from that outgrowth of the foregut, the respiratory system. Then you'd have your esophagus, stomach, and intestines. And only the upper portions of the intestine, most of the small intestines, I guess, will be included there, but anyway, portions of your intestines that follow the stomach. In addition to this, an outgrowth of the intestine during development forms your liver, gallbladder, and pancreas. Liver, gallbladder, and pancreas as an outgrowth of that intestine wall there. So these things are derived from foregut, the outgrowth of the foregut. Next would be midgut. Next portion from gut. From the midgut, basically the only thing we get there are more intestines. And that's where we have the attachment to the yolk sac there. So the midgut is attached to the yolk sac. We'll put that in parentheses so you'll know that's associated there. And then finally the hindgut, the very back part from endoderm, what's derived from the hindgut region, the posterior intestinal portions. Posterior intestines, or lower intestines we usually say, and things derived from the allantois, which is also an extra-embryonic membrane, and at least in part, but part of it is retained inside the body and becomes the urinary bladder. The urinary bladder, I'll just put bladder in there. And then beyond that, there's the cloaca. Kind of running into my other words here. The cloaca, and from the cloaca we get parts of the urethra and lower urogenital, put lower UG, urogenital components in there. So these things are derived from endoderm in the final anatomy. Now, as we go through the course, we'll be talking about different parts, and we'll make reference to which parts are derived from endoderm, mesoderm, and so forth. So these are your endodermal derivatives. We have the yolk sac and the gut separate. The gut is connected at the midgut to the yolk sac. Things from the foregut include pharynx, respiratory, esophagus, stomach, and in the upper intestines, liver, gallbladder, and pancreas, which I erased there. From the hindgut, we get posterior portions of the intestine and the allantois. Allantois becomes in part the bladder. The cloacal region in the back of the gut forms the urethra and lower urogenital systems. And part of the intestine, actually, the lowest part of the intestine. Now for our mesodermal derivatives, remember that when our mesoderm first formed, we had a central region that formed the notochord, and then beside that we had the lateral mesoderm, which segregated into a somite, next to the notochord, then intermediate mesoderm, and then somatic and splanchnic layers with a coelom in between. Parts of that lateral mesoderm going out here, and the somatic layer becomes a portion of both the chorion and the amniotic mesoderm, and then the splanchnic layer here becomes an outer covering for the yolk sac endoderm and the allantoic membrane. So those are the tissues we're dealing with here when we start looking at this. We're going to begin by just listing the mesodermal derivatives, starting with notochord. It's the most obvious first thing to think of. And then we have what we might consider lateral mesoderm, so to either side of that notochord, our lateral mesoderm is divided into that portion that becomes the somite. And from that somite, we get a few things to form. We get the axial skeleton, we think of the bases of our ribs and our vertebrae and some of the other skeletal structures along the axis of our body. Muscle and connective tissue, I'll abbreviate CT. Connective tissue, maybe I shouldn't at this stage. We'll go ahead and write in connective tissue. Muscles and connective tissues of the axial region as well, derived from the somite. The dermis of the skin, that dermis layer underneath the epidermis. Remember, the epidermis was of ectodermal tissue, and the dermis just beneath it is of mesodermal tissue. Next, the intermediate mesoderm. Spell intermediate. Intermediate mesoderm becomes mostly urinary and reproductive system features. So we have the kidney, we have ureters, we have the reproductive, well, I need to put in here gonads, I guess. Gonads, whether male or female, testis and ovaries derived here. But then the ducts that connect to the gonads in either male or female. In the male, it's what we call a mesonephric duct. Mesonephric duct. In the female, it's called the Mullerian duct. These are separate ducts, not the same duct. So mesonephric ducts, I should pluralize that, I guess, and Mullerian ducts. These things are derived from that intermediate mesoderm. And then finally, there's lateral mesoderm. Which divides into our somatic portion, somatic mesoderm, that upper layer, and from that we get what we call a parietal pleura. It's a membrane that lines our body cavities. So to the outside of your lungs are set in a cavity. That wall of that cavity has a parietal pleura there. I should say just parietal membranes would be a better thing to put here. You have a parietal peritoneum around your abdominal organs, lining that space, and around your heart you have a parietal pericardium. So all those parietal membranes and all those locations where you have cavities inside the body, those are from lateral mesoderm. Let's see. Splanchnic mesoderm. Splanchnic mesoderm lies against your organs inside your body cavity, and these are called visceral membranes. So on the outside of your liver, on the outside of your stomach and intestines and other organs in your cavities there, you have a layer of membrane that's derived from the splanchnic mesoderm. Also adhering to your inner organs, so like your esophagus, your stomach, your intestines, and so on, there's a layer outside of the endodermal component that makes up the connective tissue, the muscle, and the blood vessels that support that organ. So the muscle of your stomach that makes it churn, or the muscle of your intestines which moves food through by peristalsis and so forth. That muscle and connective tissue associated with that, and all the blood vessels associated with that, are from splanchnic mesoderm. So we're going to call this visceral muscle, connective tissue, CT, and what did I say, vessels. So all of these things are derived from those bits of mesoderm right in through there. The notochord eventually gets kind of closed up in your intervertebral disc and becomes essentially nothing in the human anatomy, nothing to talk about. In other more primitive forms of life, you might retain more notochord into adulthood, but not in most mammals. Lateral mesoderm, from that you get that somite block to either side, which becomes part of your axial skeleton, muscle and connective tissue, dermis of the skin. Remember that in the head we had head mesenchyme forming basically the same kind of material. The skeletal, connective tissue, and so forth, and even the dermis. Next, the intermediate mesoderm forms your urinary and reproductive system parts in there: kidneys, gonads, ureters, mesonephric ducts, Mullerian ducts, all that from your intermediate mesoderm. We'll see that all later when we deal with your genital system. Lateral mesoderm, we have the somatic and splanchnic layers. That somatic layer is on the outside of your body cavities, the splanchnic against the organs on the inside of your body cavity, forming the visceral membranes, and associated with those visceral organs you have muscle, connective tissue, and blood vessels that are supported there. Now beyond this we have the extra embryonic mesoderm that was associated with these different extra embryonic membranes. So from the somatic mesoderm, remember, you had the somatopleural membranes, the amnion and chorion, and from your splanchnic mesoderm you had your splanchnopleural membranes, the yolk sac and the lantois layers in there, so don't forget those. So this gives us a first view of how our embryonic ectoderm, endoderm, and mesoderm start to form the tissues that develop into our organs that we know throughout the body.